Thanks very much, Reese. My name is Mike Cousin. I'm the chair of the Scientific Advisory Committee of Epilepsy Research UK, and I'm going to chair the first session, which is on the research process, lab-based research. First up, we have Professor Stephanie Scorge from UCL, and she'll be talking about understanding animal models in research. Professor Scorge is head of the Research Department of Neuroscience, Physiology and Pharmacology at UCL, and has been there since 1999. Before then, Stephanie obtained her PhD in neuroscience at Brown University in the USA, and her research interests include gene therapy for neurological disorders and ion channels in development and the treatment of disease. And I'll hand over to Stephanie.
Okay, if all is well, you can hear me loudly. Thank you so much for coming today, and thank you for this opportunity to tell you about some of the animal models in epilepsy, and indeed how some of the feedback from people like you and from the SHAPE network has already had an impact on research that we do. So as Professor Cousins has said, as Mike has said, I do research into epilepsy and I use animal models. I am also head of the Animal Ethics Committee at UCL. So I also overview the ethics of using animals in research. And so I'm going to talk a little bit about both aspects. The first question that many people have is, why do we need animal models anyway? Why don't we just create new drugs that do what we need them to do? What's the purpose of the animal models? And in order to illustrate that, I'm going to give you an example from a field that, alas, does not have good animal models. And this is a title from a paper published a couple of years ago with a plea from a different field, the poor old people in Alzheimer's field, where they have terrible animal models. So if you think about Alzheimer's, the main thing is you lose your memory. And people struggle with that, and it progresses, and it's real cognitive decline. There are other things, but the main feature we think of with Alzheimer's disease is cognitive decline. And there are no mice who show that cognitive decline, no matter how hard people try. They have elements of it, but not very good. So the main symptom is loss of memory, but cells in a dish, very hard to show that they're losing their memory. Cells seem to remember quite well. And the mouse models also don't show memory loss. So think of your poor scientist who's trying to cure memory loss and goes into the lab, and the mice don't have memory loss, so they can't show that they're impacting memory loss in the mice. There's no memory loss to treat. So what do they use? They use proxies. So in mice, what they can show is there are other features. So plaques and tangles you've heard of. So they show in the lab, look, I give my favorite compound and the plaques and tangles go away. I'm going to try this in people. And as you will remember, if you follow the news, a lot of these treatments, when they move into human patients, they may well deal with the plaques and tangles, but often they do not help with the memory loss. And so that's a real challenge. And without that symptom that has face value, without that model that shows that you're impacting the symptom that you're interested in, it leaves your drug development slightly unguided. So in epilepsy, we're enormously lucky because we've had good animal models. And now recently, we have a growing number of animal models. So we're not just modeling epilepsy, a monolithic thing. We're modeling epilepsies, so different epilepsies. And increasingly, we're developing models that represent particular syndromes that affect small groups of patients. So it's possible to record EEG in animals. So this is a rat that has a subcutaneous transmitter that's recording his EEG as he goes about his business. And we can see seizures. And these seizures can have good, what we call in science, face value. They have good face value. They look like a seizure. If you're a clinician, I'm not a clinician, but I know clinicians, and they tell me that the seizures they see in these animals look like the seizures they see in patients. So we can treat those seizures. I would have stopped there 5 years ago. I would have said, okay, we're treating the seizures, and then I would have carried on talking about seizures. But working with people like you, working with the SHAPE network, we now, people like me who don't know as much about epilepsy, I don't have a personal experience of it, but I now understand that seizures is only one aspect. And in many cases, in some cases, not even the most troubling aspect. So we now, with feedback from people like the SHAPE network and these PPI efforts, We know that seizures are only one aspect, and there are other aspects, and we have incorporated those into our research. So now when we treat an animal who has seizures, who has a model of epilepsy, we also ask, are there other aspects of this animal's behavior? So learning and memory, anxiety, and this is a little mouse who's been put in a big open box, and if you're a mouse, that's a very anxiety-producing thing. I can imagine I myself would be anxiety. And so we look and we see, does our treatment stop seizures but make the mouse more anxious? Does it affect the animal's memory? Does it help? And this allows us to look at more than just, are we stopping seizures? So, and it is working. We are developing treatments. So for many years, anti-seizure medicines have been tested in animals, and we have a good success rate of things that are successful in animal models translate into humans. For many years, we've had a limited number of animal models, and so we kept testing and finding the same type of epilepsy, the same treatment. And so you've all heard that there's a lot of treatments that target the same pathway. That is changing because we're now developing new mouse models so we can probe, in particular, genetic epilepsies, so particular genetic epilepsies that maybe have different targets, different receptors, different treatments. And you can't develop those different treatments if you don't have a mouse model. And that is opening new ways to treat these mice as we're finding new things that can trigger seizures. We're finding new targets for stopping seizures. And we're also looking for treatments, and this is just an example of a treatment of a gene therapy we inject at one point, and the treated animals have fewer seizures, the untreated animals have more seizures. That's great in that model, but we've had to go and show that it works in a model of cortical epilepsy, it works in a model of temporal lobe epilepsy, and now we've got it working in a model of developmental epilepsy. So we're looking for treatments that can go across models, but we're also interested in treatments that work in individual models. So a treatment that is defined by, if you have Dravet, this treatment is a bespoke treatment that's looking at what's causing your epilepsy. So I can't talk about research in animals without talking about the ethics of it, because this is a hugely important topic. And what I have to say is that in the 30 years that I've been in the UK for 25 years, but in the 30 years that I've been active in research, there has been an enormous sea change in how we deal with animals in research. And partly this is because researchers are we know that if we are working with healthy animals, the research is much more reliable, it's much more reproducible, but also it's because researchers tend to feel a lot of empathy for our animals. We work with them, we care for them. So for us, looking at the pictures that we all see when you see animal rights protests, those are generally taken in the 60s and 70s, and those horrible pictures are as offensive to us as they would be watching, a racist film from the 50s and 60s is to a modern audience. So we look at those images of animals in distress, and we think there's no way that that would happen in my research group. In fact, we work hard to ensure that our animals are cared for, that they're safe, that they're comfortable, that they have toys, that they have wheels to play on. And we also have, at universities, we have a separate person at every animal facility whose job it is to make sure that researchers don't just say they're taking care of their animals, but that if any animal looks distressed, that person has the right to step in and tell the researcher, you need to stop whatever you're doing, you need to make sure this animal is safe and taken care of. And that person has absolute authority over all the researchers, and that person answers only to the welfare of the animals and the Home Office. So we have these fallbacks, and in fact, as head of the AWERB, the ethical board at UCL, I'm really proud to say that we not only are walking the walk, we're talking the talk. We are inviting groups of people through to visit our animal facilities, to see for yourselves that the animals are well taken care of, that they're happy. In fact, when we bring people through, one of the most common issues is two male mice fighting. And you have to say, look, we can't stop that. That'll last. So we do have an age limit because small children banging on cages is very stressful for mice. So we do have an age limit on that. So the future, where are we going? So most therapies We're going to have a talk in a few minutes on some of the incredible things you can do in cells to work out what causes seizures, and also the mechanisms, so the mechanisms of different epilepsies, and also how you might target those mechanisms. And so most therapies these days spend years, if not decades, working in cells and dishes to work out what's going on. When you have found your cause and you have a possible compound, and you kind of think, you know, this pathway, this particular receptor seems to be driving the seizures, and this compound looks like it's hitting that receptor, and in cells it looks like it's fixing it. That's when you can go into your in vivo animal model, and you have your hypothesis, and you have a really good idea that this is going to work in this particular model. And that's when you go in, and about 90% of the time that is done in mice. The remaining vast majority is done in rats. Rats are bigger, more expensive, just because in most research buildings space is limited, so they need bigger houses. So we have limits to how many rats we can do. And what this means is, because more and more looking at these cells, looking at these mechanisms, we're not looking at the seizure, we're looking at the thing beneath the seizure, the cause of the seizure. So we're not just going to stop the symptom, we are looking to stop epilepsy, to stop the cause and to reverse the cause, and to relieve as well. If by digging down and stopping at the cause rather than stopping at the seizure, we can stop all of the things that emerge from that cause, and hopefully relieve not just the seizures, but also all the different other symptoms that are associated with epilepsy. And that has come from information from working with people like the SHAPE Network, who are letting us know what are your priorities, what are your concerns, and what types of epilepsy is it that we should be focusing on in our new models. So I'm going to stop there, and I will take questions in the panel session at the end. But for now, we're just going to move on to our next speaker, who I'm going to quickly introduce. So yes, thank you. So thank you very much. And we're going to move on to the cells. We've promised you cells. So it's a great pleasure to introduce Professor Mike Cousins, who I've known for many years. He's now the chair of the Science Advisory Committee. He is also chair of the neuronal cell biology at University of Edinburgh, came down last night, and preclinical director at the Mir Maxwell Epilepsy Center. Professor Cousins, it's hard calling him Mike. He also chairs the Epilepsy Research UK Science Advisory Committee, and he is interested in the mechanisms that enable brain communication during patterns of neuronal activity. So thank you very much. And now we have the most difficult part of the day.
Okay, thanks very much, Stephanie. Okay, so yes, you were promised cells, so I'll give you cells. So there we go. So my role here really is to tell you what's the point in looking at cells. We know that people have epilepsy, we know animals have epilepsy, we just heard that. So what's the point of looking at the cell molecular level? You know, what insight can we get that's really going to help put treatments into practice more quickly? So when you think about the cell is what you can think of as the basic unit of physiology. It makes up organs, and obviously organs make up our body. And the thing that defines a cell is this thing called a membrane. This is a physical barrier that stops things coming in, and it stops things escaping as well. So the cell you can think of as a discrete entity. Now within it, it's also got membranes inside too, which make little compartments within the cell. For example, all your genetic material is stored within what's called the nuclear membrane. And that means you can think of the cell also as a whole little unit, a little microcosm within the whole body. Now cells talk to each other, but they also talk within each other, within itself as well. For example, these little compartments can communicate with each other, and that's called intracellular signaling. Furthermore, these cells can talk to each other as well. And that can either be unidirectional, or you can talk to each other bidirectionally. And that's called intercellular signaling. Now, I was a bit confused by these terms. So I always remember it by, if Scotland are playing England at football, it's an international. And if Rangers are playing Celtic, It's in the Scottish League, so it's intranational. I'm not sure if that helps or not. So, two cells talking to each other isn't going to do it in terms of a complex organism like a mammal. So cells can also talk to multiple cells at the same time. That can be done, for example, by releasing hormones into the blood, so it can hit lots of cells at the same time. Or it can be through multiple physical contacts, like synapses, which I'll come on to in a sec. So why bother looking at cells in epilepsy? What's the advantage? Well, if you had a little microscope and you zoomed in on the human brain, say up here, you would see something like this. You would see lots and lots of brain cells all connected together in these intricate pathways called circuits. And typically, the human brain has about 100 billion neurons. It's a crazy number. What does a neuron look like? Well, this is what a typical neuron looks like. And they're designed in a way that information only flows in one direction. And this slide from left to right. Information is received at these things called dendrites. It's then transmitted along this thing called an axon in the form of an electrical impulse called an action potential. And then the signal is then given out at these things called nerve terminals. And as I say, it's designed for information to flow in one direction, usually. Now, these neurons talk to each other at things called synapses, shown here. And this is the axon of one neuron coming down, and here's the nerve terminal, and here's the dendrite. A typical neuron has around 1,000 synapses. And if you remember, you have 100 billion neurons in your brain. That means there's approximately 10 to the power 14 synapses in your brain right now. That's a ludicrous number. It's what gives the brain its computational power. It's also amazing that actually we're all sitting here and we're pretty much doing okay because there's so much propensity for things to potentially go wrong. And of course, things occasionally do go awry, which is why some of us have epilepsy. However, there's an issue actually with the synapse, and that is, you can't really see it here, but there's a physical gap between the nerve terminal and the dendrite. And electrical signals can't jump physical gaps. So how does that work? Well, imagine you've got two neurons who want to talk to each other. Information should be flowing in this direction. What happens is the first one gets activated at this dendrite. This electrical impulse comes down this axon. And when it reaches this nerve terminal, it releases chemical neurotransmitters. These chemical neurotransmitters then diffuse across the synaptic gap. They bind to this dendrite, and then this propagates an electrical impulse in the neighboring neuron. And that's called electrochemical transmission. And it's the basic unit of currency that all neurons communicate with each other with. Now, how do these chemical neurotransmitters get released? Well, that's down to these things called synaptic vesicles. There's typically about 200 or 300 in a typical nerve terminal, and they're crammed full of these chemical neurotransmitters. And when the electrical impulse invades, these things fuse with this impermeable barrier or membrane to release the neurotransmitter content. Now, the neurons have an issue because, as I say, there's quite a small number of vesicles to keep up with the demand for communication. So what the neuron does is it remakes these vesicles locally to make sure there's always a continuous supply inside this nerve terminal. And that's done by these processes called endocytosis. I'm not going to go into any details here, but endocytosis remakes these vesicles to make sure we've always got enough for releasing neurotransmitters. Okay, so what I've told you so far is really just a very basic summary of how brain cells communicate with each other. However, what I've not told you about is how this links to epilepsy. So if you'd said to me, or anybody in our field, about 10, 15 years ago, defects in neurotransmission would underlie certain forms of epilepsy, I think most people would be quite surprised, because this is such a fundamental component of brain function. And if anything is messed up in that, you think that would be really problematic. But it actually turns out that's not the case. We've got essential proteins for vesicles fusing to release transmitter, like VAMP2 and syntaxin, and also essential proteins that remake vesicles, like dynamin-1 and clathrin, that are all found in people with epilepsy. So I'll finish there. I just want to thank one person in the lab who did all the work on the dynamin mouse model. That's Catherine Bonnycastle, who unfortunately left us 6 months ago to be a genetic counsellor. We've now got a replacement in, so hopefully we'll make Good progress again. And that. Okay, thank you very much. And again, I'll take questions at the end. Okay, so next up, we have Gareth Morris, one of our new fellows, who was awarded a ERU, Epilepsy Research UK Emerging Leader Fellowship Award in 2020. And really looking at a brand new way to try and treat epilepsy using gene therapy. He's a senior research fellow at the Department of Neuroscience, Physiology and Pharmacology at the University College London, and he'll be speaking about Batman in the Brain, gene therapy for seizure prevention. Gareth.
All right, thank you very much, Mike. And can you hear me okay through this microphone? Okay, I'll just try to stay put here and skip through all of Mike's beautiful extra slides. You just wanted to make this as slick as possible. Right, that is us, and that is me. Yeah, so thank you very much to Mike for the introduction, to Epilepsy Research UK for giving me the chance to come and speak, and obviously to all of you guys, the Epilepsy Research UK SHAPE network, whose input to our work is so valuable. My name's Gareth. As Mike said, I'm an Epilepsy Research UK fellow down at UCL, just down the road. And it's very exciting to talk to you all this morning. This is actually the first time I spoke to a group of real people in the last couple of years. So yeah, it's a real pleasure. So I'm going to talk to you this morning about gene therapy, which is sort of what we hope will be a treatment of the future for epilepsy, the reasons why we think that that might be a really good thing, and we'll touch on a couple of possible limitations we're trying to overcome. And I've been tasked with explaining that through the medium of Batman. So good luck to me. So first of all, let's talk about why we actually need gene therapy. We have treatments for epilepsy, we have medications, so why are we still doing this research? So I don't need to work through these infographics of what is epilepsy for this audience, but just to mention, epilepsy, it affects people of all ages, and there's a whole host of different possible causes. So it's definitely touched on. Epilepsy is not really one brain condition. It's actually sort of an umbrella term for a family of different things with different causes. And this already presents us a challenge in trying to treat that and address it. Now, as I say, we have anti-seizure medications, and these work well for about 70% of people whose seizures are resolved by medications. But this clinical picture leaves a few challenges and issues which we're trying to resolve. So what are those challenges? Well, first of all, people that have epilepsy may have to rely on daily medication. Now, what happens if you forget to take your medication, for example, or you don't have access to it? That becomes a big problem. So if we can create therapies which are longer lasting, that might be very helpful for people. Now, as I mentioned, 30% of people with epilepsy will still continue to have seizures, even with the optimal medication profile. And so we're really trying to create new things which will help those people to stop having seizures. Furthermore, from the conversations that I've had with people that have epilepsy, some of these medications, even though they might stop seizures, as we say here, they can have quite nasty side effects and can create challenges with things like memory, with things like sleep, things like psychological health. And so we're really trying to create things which might not cause those side effects. And now, what I'm going to sort of dwell on quite a lot in my talk, a lot of the reasons for this is that the current medications lack specificity. And you'll see more what I mean by this, but what I want you to have in mind, and I appreciate this talk is slightly biased towards focal epilepsies, but we can talk about genetic epilepsies during the discussion at the end if people like. What I mean by this specificity is that often seizures arise from a very small part of the brain. But when we give these medications, they actually affect the entire brain. So they might stop that little bit of the brain that's causing seizures, but they will have impacts on the rest of the brain, which we want to try to stop. So what is our research mission? What is the research mission of my lab and labs like Stephanie's lab and Mike's lab? Well, we want to create ways which will reduce the excitability of the brain, which is causing seizures. So seizures are caused by overactive circuits in the brain, but we want to do that without having any impact on the rest of the brain's goings on and functionings. And gene therapy, we think, is a good way to do that. So in terms of the specificity, what does an ideal treatment for epilepsy look like? Well, first of all, as I mentioned, we'd love to develop something which is very long lasting or even permanent. So, you just have to take this thing once and then you can kind of forget about it. You don't have to keep taking medications every day. Now, I mentioned within the brain that we want to treat just that bit of the brain that's causing seizures and leave the rest of it as it is. So a better treatment would actually be spatially restricted, we call that. It will just hit the seizure focus. Now, Mike very conveniently introduced all of these sort of circuits and connections between the cells in our brain. And as you can imagine, they're very complex. And as it happens, within this seizure focus, not all of the cells in our brain are actually involved in causing the seizures, only some of them. So if we can further specify our treatment just to those cells that are causing the seizures, again, we can try to leave alone the normal brain functions. And then finally, we'd love a treatment which gets activated only when seizures are actually likely to happen, and it's just kind of switched off for the rest of the time. So that means, most of the time, our therapy would not be doing anything at all, but it's kind of sat within the brain. When it's needed, it can switch on, hopefully stop seizures from happening, and then switch back off again. So this is where we bring in gene therapy, because we think that it goes some way to addressing these questions. So now, what is a gene therapy? It probably sounds a little bit scary, right? Well, it all starts with viruses, and I appreciate viruses are not flavor of the month for the last couple of years, so I'll try to win you over. So this is actually called a viral vector, and this is what we use to deliver our gene therapy. Now, there are two parts to this which are very important. There's a packaging protein around the outside. This is sort of these little prongs with balls on the end, if you like. This is what's going to help us to actually put our therapy into the cells. And then in the middle of that, we have the virus's genetic material. And this is something that we have complete control over. So we can design whatever we want in here. We can take advantage of the ability of the virus to get into cells, and we can sort of exploit that to deliver what we want. It's kind of like a Trojan horse, if you imagine. And just to reassure you, these viruses have been quite ruthlessly stripped of anything that can help them to replicate. So they're not contagious, they're not going to develop or spread within the body, they're just going to go in and sort of sit there and express this gene. Now what's nice about that, first of all, because it will just sit within our cells, it's very long lasting. We would predict that these would basically be permanent, and gene therapies in Parkinson's disease have been shown to last for at least 15 years. Secondly, how this would work is if you have a brain here and this, let's imagine this is a seizure focus roughly within the temporal lobe there, we can inject this thing directly into the seizure focus and we can resolve just the activity within that little area. And because these viruses are relatively big, they're not going to spread too far within the brain. And that means all of the rest of the brain here will never see this virus, will never be affected by it in any way. So this is that sort of spatially localized treatment that I wanted to get across. And as I say, the second point that we wanted in our therapy is that it's limited to the seizure focus. Now, if we zoom in a little bit, I won't bore you with too much virus genetics, but as I say, we can control what is actually expressed in this thing. And in very simplistic terms, we're going to have a helpful gene here. We don't have to worry about what that is. It could be a number of things, but it's going to be something which stops seizures. And just before that, we have something called a promoter. Now, in order for this gene to actually be expressed in a given cell, that cell needs to also have this promoter. And some promoters are quite ubiquitous and broad. They're expressed in a lot of different types of cells. Some promoters are very specific to certain types of cells. And so we can use that to design our therapy so that it is only expressed in certain types of cells. So Mike very nicely did this more eloquently than I can, but if we zoom in on a particular part of the brain, what we see is these individual neurons, and as Mike said, they have information flowing between them. And let's imagine we have two different types of cells. We have our sort of pinky, purple, whatever color that is, and we have these orange ones here. These cells might have different types of promoters, and this promoter A, this pink one, might only be in the pink cells, and promoter B, the orange one, only in the orange cells. And what I want to get across is that if we've delivered this gene into this group of cells, it's actually only going to be expressed within the pink ones and not the orange ones. And so in that way, we achieve this point number 3, that the treatment that we give is limited to specific types of brain cells. And this is actually a pretty good overview of the current state of the art of gene therapy. And so I'm just going to show you some real data, which has been led by Stephanie. So you can see here a very nice academic neuroscience publication from a couple of years ago now. And what Stephanie's group did was take, so this is the viral gene that was delivered. Don't worry about the different components, but what you have here is a promoter, like I just explained, which limits the expression to the certain types of cells that we want to target. And you have this therapeutic gene here, which will reduce their activity, and also a little pretty fluorescent marker so that we can see where these things actually are. Stephanie's group used an animal model, as she's described. And you can see here, this viral construct has been injected into a mouse hippocampus. And we can visualize this fluorescent protein here using microscopes. And you can see that the treatment's limited to the hippocampus, which is often what we want to target. And if we actually count the number of seizures experienced by these mice, they're hugely reduced by this treatment. And you can ask Stephanie more about this if you like in the questions, but this approach will begin trials in humans, I believe, later this year. Stephanie's tentatively nodding at me. So this is, becoming a clinical reality, everything I've described to you. But if you remember, I've discussed 3 advantages of this, but there was actually a 4th that I described. And this is the focus of my research, which is trying to develop what we call on-demand gene therapy that's only activated when we think it is needed. So as I say, a better treatment would be active only when seizures are likely to happen. This is where Batman comes in, right? So I'll try to explain this. I'm not a Batman expert, so forgive me if I mess this up. So Batman lives in the city of Gotham, right? He's a superhero. He runs around in his cape. And, in normal times when Gotham is safe, Batman just kind of lives in the shadows and you don't see him. He's not doing anything. When there's danger, when there's some sort of super criminal, the city of Gotham shine this bat signal into the sky. Batman will see it, and he will rise into action, go and capture the bad guy, and Gotham is safe again. And this is kind of like what we want to achieve with gene therapy. We want to have the gene therapy as our Batman always sat within the brain, always there. But just as you don't want a man in spandex running around your city all day, every day, you don't want your gene therapy to necessarily always be active. And so what we're trying to do is create something that acts like Batman and responds to some sort of signal going on within our cells in a seizure. So that is all from me, more or less. And now we'll move into a panel discussion with all of the speakers. And so you're welcome, obviously, to ask us any questions. And I'd like to just close by asking you a couple of questions. So, as Stephanie points out, it's very helpful for us to hear from the SHAPE network and people that live with epilepsy about how our research can actually help. So we'd love to know, what do you think about gene therapy and what I've just showed you? Would you be willing to use this? And what concerns might you have about that? What would they be? And how can we help you to address that? So, tell us, use the SHAPE network, and help us to shape epilepsy research. Thank you.